Hello, welcome to the enzyme investigation, where you'll have an opportunity to see how enzymes work, as well as how well they work in different environments, like pH, changing the temperature, changing the concentration of the substrate, or changing the concentration of the enzyme. So for this particular lab investigation, we are using an enzyme called catalase, which is found in living tissue. And for this lab, we are going to use red potatoes and actually grind them up with water in a blender and strain them into the beaker. We're also going to use a substrate that's pretty common, you may have heard of before or used before. It's hydrogen peroxide. 3% is what we're starting with. And then we're also using distilled water. So for the potato, generally, we want to use a red potato in this situation to get hopefully the best results for our enzyme. So we're going to take cut up red potatoes as our source of catalase. Remember, catalase is an enzyme found in living tissues, even our own tissues. And we're going to put that in the blender. And I previously cut off all the skin because we're really wanting just the catalase, which is found in the actual white tissue of the potato. And then I put in about 200 milliliters, doesn't have to be an exact amount, of water. We're going to put that on the blender. And then I'm going to go. Blend that up, about 30 seconds. And then what I do, you can either pour this through cheesecloth or a strainer just to get all the little bits out, because we just want that catalase concentrate that we're going to put into our test tubes later on in the experiment. So the catalase solution is draining into our large beaker, and this is going to be the source of the catalase that will go into the test tube eventually and react with the hydrogen peroxide as our substrate. And the hydrogen peroxide, when it gets broken down by the catalase, will turn into two products, water as well as oxygen gas. And the oxygen gas is actually going to form white bubbles, and the height of the bubbles, or foam that we sometimes call it, will actually be measured in order for us to see how well the enzyme is working to break down the substrate. And that will be, for all four variables, our dependent variable. What we're going to change, depending on which variable you're focusing on as your independent variable, will be things like temperature, pH, the concentration of the catalase, or could be the concentration of the substrate, which is the hydrogen peroxide.